Today we're going to talk about how to take DNA and read it to make a trait. So is that trait your hair? Is it your eyes? How tall you are? How round you are? How big your feet are? All of these are your phenotypes. And they're based on your genes. A gene is only a portion of DNA. Not all of your DNA contains genes. And so to read DNA, there are two parts, and we're only going to talk about the first today. DNA must be copied. And so I'm going to create a piece of DNA here, and I've opened it up a little bit just so we can see better. We already know that there's base pairing rules for DNA, and we know that this is how it works. I'm just making the code randomly. So what we do is we take a protein called RNA polymerase, and it is going to copy that DNA one letter at a time. To make a new strand. So the strand I just made there is this. Notice we only copied one of the two strands. The strand of DNA that we copy is called the template strand. The other strand is called the coding strand. We do not use the coding strand, we use the template. And so the template strand, as a reminder, up here was T, A, C, G. And so you might notice that in RNA there is a U instead of a T. Over here we had a T where there was an A, but RNA is a U. So that U is a new nitrogen base called uracil, and that is only for RNA. That is one way the cell tells the difference between DNA and RNA. The other difference is in the sugar. So let's look at that real quick. DNA nucleotides look like this. Whereas RNA nucleotides look like this. Almost the same, except here we have an oxygen and here we do not. That is why we called this sugar deoxyribose. Deoxy means without oxygen. Whereas this sugar here is just ribose. That is why the first letter of RNA is R and the first letter of DNA is D. They are both nucleic acids. So those are the big differences there. Another difference is that DNA is double-stranded and very big, whereas RNA is single-stranded and rather small. Let's see, what else do you need to know? When we make this RNA here, that's not the end of the story, so if I were to make this bigger, there are parts to this that we need to get rid of, and those are called introns. And then the parts we want are called exons. We cut this stuff out, glue together the exons, and they leave the nucleus to be read. And so transcription is just taking a piece of DNA and turning it into a piece of RNA. We usually call that RNA mRNA because it is a message that leaves the nucleus. Once it leaves the nucleus, it will be used to make a protein eventually. And that protein is your trait. Let's watch a quick video on sort of how that works. Here's a great video of that happening.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left.
All right, so what are we looking at? This is DNA. Down here somewhere is a gene. This right here is called a transcription factor. It is essentially waiting there for RNA polymerase to bind. It's like little hands reaching out. Come here, RNA polymerase, I want you. And so eventually this will come to the transcription factors. These transcription factors are important. They attach only when the time is right for transcription to occur. It is how our cells interact with our environment. And so, for example, if the sun is shining, that might say that the melanin gene needs to be read. And so transcription factors will bind and it will be read. Watch how it grabs RNA polymerase. There it is. And it's got it.
The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2...
Okay, so what you're about to see here, this is called an enhancer. And this loop here is going to be called a gene switch. That switch goes off as another level of control to make sure that we're not always expressing genes. Because you have a lot of DNA, there'd be no point in always expressing all of the genes. So this ensures that we only express the ones we want to when we need to.
2, 1, go. The blue molecule racing along the DNA is reading the gene.
Alright, so this is a great image here. We've got DNA right here, which has 2 strands. Only one of those strands is being read, and that is the template strand. Right here, this growing strand is RNA, which is only single-stranded. These are all little RNA nucleotides. They're being taken in and making this strand grow. The RNA polymerase is going this way, downstream. Downstream means towards the gene. And we are executing transcription.
It's unzipping the double helix and copying one of the 2 strands.
Only one of them.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole.
And so, remember, it's only a cousin. So this RNA here has that oxygen, whereas this DNA down here does not. That is why this is deoxyribose, while this is just ribose. So the sugar here is different, but also on this we have uracil, whereas down here on the DNA we have thymine. And so there are no thymines on these nucleotides.
They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Yeah, all right. Let's see. Just wanted to reiterate, if there's an A in DNA, we add a U instead of a T. And then I also wanted to just point out, once we've made RNA, we have to get rid of those introns to make what's called a mature mRNA. That then leaves the nucleus. Here's how we do it. We splice. So if this is an intron, we get rid of it by making a loop, and then we cut it out that loop. And so a little loop floats away, and we recycle that. All right, in the next part, we're going to talk about how we read RNA, 3 letters at a time. Using those 3 letters, we're going to bring together ingredients to make our proteins, and that happens next.